Hello everyone. So we are continuing with the chapter cell cycle and cell division. So already in the previous video we have understood cell cycle, but we have just understood the interphase. Properly we have understood interphase, and we know that interphase is divided into G1, S and G0 phases. And G1 phase can also, the cell can come out from G1 phase and it can rest for some time, and that phase is called G0 phase. So everything in detail we have already done, so we will not waste our time in explaining and again or discussing that. So we will directly jump to mitosis. But before that, I would just like to tell you one thing which is usually confusing for some students, that in NCERT questions have been asked based on this. Like for example, if this is a cell cycle, they will just ask you, now in this cycle you know that this is mitosis phase and the rest all is interphase, and here also we have G1, yes, and G2. Okay, now the confusing question which is actually asked based on this concept is about that amount of DNA as well as haploid and diploid. Usually you know that the cell which is diploid, if the cell is diploid, it will divide into two cells, okay, which will also have diploid number of chromosome. That means if a cell is having, for example, any chromosome number, like if you take human beings also, it is 46, diploid number is 46, and then here also it will be 46 and 46. Now what kind of question will be posed on this concept is that they will ask you, when the cell enters the cell cycle, okay, then they will ask you what will be the number of chromosome in the S phase. This will be the question. What will be the number of chromosome in S phase? See, whenever cell has entered G1 phase, here also it will be 2N. When it passes G1 and comes to S phase, even its DNA increases in number, increases its amount, there only DNA increase has happened. Replication of DNA has happened, but chromosome has not doubled. So here also it will be 2N. Okay? And when it crosses G2, here also it is 2N. When it comes to M phase, here it actually divides into two cells. Okay, here also the cell was 2N. Actually here it divides into two cells, and also it will have 2N and 2N. So there is no any kind of doubling of chromosomes. No, there is no doubling of chromosomes anywhere in the cell cycle. Only thing you should remember, that the content of DNA, when we are talking about the content of DNA, then what you need to remember is that when the cell divides, okay, now, so what you will understand here, and this is about the haploid and diploid cell, okay, set of chromosomes, two sets are there. But when you talk about amount of DNA when the cell enters the cell cycle, it will be 2C. Because you know that the amount of DNA in haploid cell will be considered as C, and the amount of DNA in diploid cell is considered as 2C. That is why it is 2C. Once it enters S phase, the actual replication will happen. That time what will happen? From 2C it becomes 4C. Only the amount of DNA has increased. Chromosomes have not increased. Okay. Then after that what will happen? Once it reaches the G2 phase, here also what will be the number of amount of DNA? Here also it will be 4C. Okay. And here also it will be 4C. And the cells which are, here it is 4C. But once its division is done, these daughter cells will have 2C and 2C. Clear? Because you know that division is done. Amount of DNA was doubled because it was needed to be divided into two daughter cells. So that is why it became 2C and 2C again. So questions will be posed on this. They'll ask you. They will give you some amount of number of chromosomes. They will tell you. In NCERT you might have seen, there is one example about onion. Okay, so onion cells, usually they have diploid chromosomes, that is 16. So they are asking you, in the cell cycle, when the 16 will increase? No, 16 will not at all increase. Completely undergoing the cell cycle, the onion cell will have 16 only. Okay, because it is mitosis. If it was meiosis, then reduction of chromosome would have happened, but it is not meiosis, it is mitosis. So chromosome number will not increase nor decrease. What increases in S phase is the amount of DNA, and it is always from 2C to 4C. Clear? I hope I have made this also clear. Okay, you can take down this much. Okay, so now we will directly jump to the M phase of cell cycle. So we have already completed interphase properly without any confusions. Everything is clear. So what we will start now? We will start with M phase. What is M phase? M phase is mitosis phase. So in mitosis, what will happen? You know that if this is a cell, okay, whatever the number of chromosome it has, when it divides into 2 cells. And these cells also will have 4 and 4. This is mitosis. This is, you can remember it like, you have to have this diagram in your mind for mitosis, and the same diagram, this type of diagram is there for meiosis also, which I have already told you in the previous video. So that also you keep it in mind. It becomes very easy to understand all the concept, okay, without any kind of problems. So this is in mind, you can remember it, okay. This is a mind map. Now let us consider this now and find out what all will happen, what are the stages of mitosis. So let us start. First of all, we are talking about mitosis. Right? So mitosis. What is the other name for mitosis? Yes, mitosis is also called as what? Mitosis is also called as equational division. Why it is called as equational division? Because it is going to give the same amount of chromosome, whatever it has, whatever the parent cell has. So we call it as equational division. And mitosis is divided into 2 important phases. Already we have discussed that. Which are those 2 important phases that we will see now? The 2 important phases are like this. Okay. First of all, it is called karyokinesis. Okay. And then another one is cytokinesis. So first one is karyokinesis, and the second one is cytokinesis. Clear? What happens in the karyokinesis? The nuclear content is divided into 2 equal parts, or the nucleus divides into 2 equal parts along with the chromosome, along with the DNA. What about cytokinesis? It is the actual cell division where cytoplasm also will divide and then 2 cells are formed. Okay. Now, we have to discuss about everything in detail. So, we will see the different stages of karyokinesis. Now, karyokinesis, as soon as the G2 phase ends in the interphase, the next phase only is karyokinesis. And which is the first stage in this karyokinesis? We will see that. So, one by one we'll be seeing here which are those steps of karyokinesis. So, the first step in karyokinesis is called prophase. It is called prophase. And the second stage is metaphase. It is metaphase. And then we have anaphase. And then we have telophase. Clear? So, once the cell has started the karyokinesis, the first stage of karyokinesis is the prophase. As soon as prophase is completed, it comes to metaphase and then anaphase and then telophase. Once the telophase is ended, then the karyokinesis will end. Then what will happen? The next important phase of the mitosis is cytokinesis, where the cytoplasm will divide. We will see along with the diagram. Before that, you just take down this much. Okay, fine. So see here, first of all, students usually, have come across some students who actually find difficulty in remembering this, the things what all will happen in the stages and to remember the stages. I would like to tell you all one thing. If you have understood the concept, there is no need to by heart anything. There is not at all any requirement of by hearting anything. If you have understood the concept, you yourself will start, writing the answers. You will know everything what will happen. So understanding it is important. So we are doing along with basics. So you have understood, you have some knowledge. What is mitosis now? Okay. So now it becomes very easy for you to understand all the stages. Okay, then let us start. You know that karyokinesis is what? It is actually the nuclear division. Already we have done about this, but still I'll be writing it is nucleus division. You can say it is nucleus division. What about cytokinesis? It is cytoplasm division. Afterwards I'll be just rubbing this, so you can take down this one. It's nucleus division and it is cytoplasmic or the cytoplasm division. Okay, fine. Now let us understand what is prophase. Okay, slowly we will understand what is prophase and we will understand all the stages now. Before understanding prophase and all the stages, I would like to give some introduction about scientists, which I forgot to do. So the scientist people who actually found out this mitosis, we have to remember them first, no? Tribute should be given to them. Okay. The first people, the first scientist was Strasburger. Strasburger, actually he found mitosis in plants. So who found mitosis in plants? It was Strasburger. What about in animals then? It was Fleming. The scientist's name is Fleming, who actually discovered double M, I guess. I think it is double M, okay, in animals. Okay, so Strasburger, what he did? He found mitosis in plants, and Fleming found that mitosis happens in animals as well. And then, who is more important? Both are more important, but one more thing about Fleming is that he is a scientist who actually coined the term mitosis. What he has done? He coined the term mitosis. So this is, you should be clear. Two scientists are there, Strasburger, who actually found mitosis in plants, and Fleming, who found mitosis in animals. But Fleming is the one who gave the name, or who termed it, who coined the term mitosis. Clear? So this was actually, I need to tell it the first, but I just forgot. You can take down this one. Okay, now coming to the stages. Okay, we'll not waste the time now, we'll just start. Fine. So before entering the karyokinesis or before entering the mitosis, the cell will be in the cell cycle phase, wherein it has completed G1, it has completed S, and it has completed G2 also. Then how the cell looks, I will be showing that here. You just remember this cell here, because you need to compare this with all the stages. This cell will have its own nucleus. When it has completed G1, S and G2, how it looks, I am showing that. Here it has its DNA. I am showing two colored DNA here because to make you understand that it has replicated, because it has crossed its S phase. Then also it will have all the other, what you can say, cytoplasmic content. Fine. And then one more important thing is, it has already what? Doubled what? The centrosome. I'm just showing you like this with red. Okay. I'm not naming it because no place properly. So first of all, what you need to remember here, the thing is, this is a cell which has completed the 3 stages of interphase. So it has doubled its centrosome. It has doubled all the cell organelles, cytoplasmic content. Also it has doubled the DNA. Okay. It is ready for the division now. So what will happen in the prophase? Before explaining you prophase, I would like to explain directly metaphase. Now you will be confused. Why is directly metaphase? Because prophase is a stage which is actually the preparatory stage for metaphase. So what all things are happening in prophase? All these things are preparation for which phase? For metaphase. So metaphase is not that difficult stage. It is very easy. You will remember it easily. So once you know what is going to happen in metaphase, then obviously you can remember this all the things which is happening in prophase. Okay. So it becomes easy for you. So I am first explaining metaphase. Meta means what? Meta means center, or you can say it is equator. Okay, now during metaphase, how the cell looks and what happens, I'll show you this. So this is the cell. I am talking about which one? I am talking about metaphase stage. Okay, be clear with this. So this is the cell, and already you saw here the cell has doubled its DNA, all the content, everything is shown here. During metaphase, what you will see is that there are two things which is important here, and I'll be showing you only two chromosomes. Okay, so this is one chromosome. Okay, and this is one chromosome. So metaphase means what? Meta means center, or you can say equator. So in metaphase region, two important functions of metaphase stage is that chromosomes lie at the center of the cell. They lie at the center of the cell. This is one thing. Now, do you think easily they can lie at the center without any support? Do you think that it is possible? No. Some support is required, no, for this chromosome to be at the center. So what is that support? Who will give that support? And that support is provided by the fibers. You know that already the centrioles have divided. You can see the centrioles here and here. These two, the red ones, they are centrioles in the cytoplasm. So these centrioles will provide, this produces what? Spindle fibers which will hold the centromere at the center. Okay. So in this picture, it's very easy here. Which are the two things in metaphase? First thing is chromosome lie at the center of the cell. Second thing is they cannot lie easily like that. So something should be, support is to be given to that. Something should hold that chromosome, and that support is given by the spindle fibers. Spindle fibers are produced by centrioles. Centrioles duplication already it has taken place in the S phase of the interphase. Now what you can see is in this metaphase stage, two things. Chromosome at the center and spindle fiber have attached to the chromosome. Only these two things are found in the metaphase stage. So now we will study prophase. First of all, you take this much and then we will study prophase. Or else we'll do one thing. I will write the characters of metaphase first of all. Okay, so I'll be writing it here. So this you can write, and the stages, the functions, I'll write it here. The first thing is chromosomes at the center. So this is the first thing. Second thing is what? What is happening here? You can see that chromosomes are attached to spindle. Chromosomes, and particularly chromosomes, the centromere part. Kinetochores, no? Kinetochore attached to the spindle. What are the two important points of metaphase? Chromosomes are found at the center. Chromosomes are attached to the spindle. Which part of the chromosome is attached? You know that chromosome at the centromere region, at the side they have this proteinaceous plate. I told you already, we have done this in morphology of chromosome. They are called as kinetochores. The spindle fiber get attached to kinetochore, which has, which is placed at the centromere region, peripheral to the centromere region. So first take down this metaphase points. Okay, so once you have understood what is happening in the metaphase region, Now it becomes very easy for us to understand what all things have happened in the prophase. So now let us study prophase. Okay, let this diagram be here. To understand prophase, it's very much important. This diagram should be here. Okay. Now, first of all, compare this cell and this cell. What are the differences you are able to see? Here you can see that DNA is in the form of thread. And here it has become chromosome. Then which stage DNA got condensed? Obviously, DNA got condensed in this stage called prophase. So what will happen in the prophase? The DNA which was in the form of thread, it will start condensing. So the first step in prophase is DNA condensation. It is DNA condensation. So DNA gets condensed to form what? To form chromosome. And you know very well that DNA condenses along with histones to form what? Chromosome. Right? So DNA condensation, that means the formation of chromosome starts where? In which phase? That is in the prophase. Clear? Now, second thing what you can compare, what you can understand, what is the difference? You are finding nuclear envelope here and there is no nuclear envelope here. That means during the prophase, nuclear envelope have been removed. Now the question arises, why? See here, whenever DNA condenses, it forms chromosome. Now you want to bring this chromosome at the center, a lot of movement should be done. Okay, once the DNA gets condensed, it will start accumulating more place and it needs more place for that function. So if it is within the nucleus and within that nuclear envelope, the space is not provided easily. So the nuclear envelope will be removed. That means the nuclear envelope will be completely removed. So the second thing what happens in prophase, nuclear envelope disintegrates. Are you trying to understand what happened? The first thing is DNA condensed to form chromosome. Why? Because prophase is a preparatory phase. It is preparing for metaphase. So DNA got condensed to chromosome. Second thing, it needs more space. Chromosome needs more space. So if nuclear envelope is present, then more space is not provided. So nuclear envelope is removed. That is, nuclear envelope disintegrates. Now you know that within the nucleus there is one more thing present, and that is nucleolus. Yes or no? And nucleolus is visible only when the DNA is in the form of thread. So once the DNA starts condensing, then you will not be able to see the nucleolus. Are you able to find any nucleolus here? No. So what will happen along with nuclear envelope disintegration starts? At the same time, nucleolus disintegrates. Correct? So these three things easy to understand now. So once you remember this diagram and this diagram, easily you can write down all the points of prophase. Now anything else are you able to see here? Yes, DNA condensation is done, nuclear envelope disintegrates, nucleolus also disintegrates. Okay, and then what you can see is the difference here and here. The main difference you are able to see that here the support, that is the spindle fibers are formed. I mean you can see here in the metaphase. Then already they have formed and they are ready. They are already connected to chromosome in metaphase. Then where they were started to form. So spindle fibers started where? During the prophase. I'll be just making it small. So the fourth step or the fourth important function of prophase is that spindle fiber formation. Spindle fiber formation. Clear? So all these are what? All the four things what I talked about is all this is related to prophase. This diagram is related to metaphase. This diagram is related to metaphase. All these things what I have told now, this is related to prophase. Okay. So I will show you the diagram of prophase also. but important is you need to remember the things. These are the most important 4 functions which are happening in the prophase. The first thing is DNA gets condensed to form chromosome. Second thing, nuclear envelope is disintegrated. Whenever nucleus is not found, along with at the same time nucleolus also disintegrates, because nucleolus you will not be able to see when DNA forms chromosome, you will not be able to see nucleolus. Then to keep that chromosome at the center, the support system is spindle fiber. Already in this phase you see that centrosome have divided into 2 parts. Now actually they start producing fiber. Where? In the prophase stage. Spindle fiber formation starts in the prophase. Clear? I hope I made it clear. Take down. Okay. Now, so I'll be rubbing this metaphase, and metaphase we have done no already. Metaphase, only thing is there are 2 functions. Chromosome at the center, spindle get attached to chromosome. Clear? So I hope prophase is easy now to understand. Now I'll be showing you the diagram of prophase so that you can take down the diagram of prophase. So during prophase, I cannot show anything properly. You have to remember it, what all things will happen. So just I can show like this, that I can show that DNA is starting to form what? It is condensing to form the chromosome, or it has not been completely, it's not complete chromosome in the prophase. That means condensation have started. Condensation will complete in metaphase only. I mean completely condensed chromosome you will find in the metaphase. But here it has started. So I'll be showing it because, okay. Anyways, just try to understand that DNA has started producing condensed chromosome like this. Okay, then what you will be able to see? Nucleus, the nuclear envelope. Actually, nuclear envelope will start what? It will start disintegrating. Okay, so this is how you can show that nucleus have started disintegrating. Nuclear membrane has started disintegrating. Are you understanding this? Then what you will find? The nucleolus, even this is disintegrating. Then what? The next thing is what? It is spindle fiber formation. It has started already. Duplication was done. Centriole duplication. Now the spindle fiber formation have started. Like this. Clear? So the first thing is DNA got condensed to chromosome. Second thing, nuclear envelope disintegrating. Third thing, nucleolus disintegrating. Fourth is spindle fiber formation. I will show spindle fiber with red. Okay. Like this, it has started forming. So this is the diagram of prophase. Please take down the diagram along with the, write down that. Fine. So next, so we have understood prophase, we have understood metaphase. Now what will happen, that we need to understand. I'll be rubbing everything now. We will just focus on anaphase. Okay. Prophase is a preparatory phase. Metaphase is a central region phase wherein chromosome at center. Ana means polar region, towards the polar region. So what anaphase means, we will try to see that. I'll show you metaphase here and we will compare it, okay? In metaphase, what we saw that, in metaphase, the cell was here and the chromosomes were at the center. Correct alva? Concept clear ಆಗಿ ಅರ್ಥ ಮಾಡ್ಕೊಂಡ್ರೆ you people don't need to ಏನನ್ನು ನೆನಪಿಟ್ಕೋಬೇಕಂತ ಇಲ್ಲ, ಅಂದ್ರೆ ಬೈ ಹಾರ್ಟ್ ಮಾಡ್ಬೇಕಂತೇನಿಲ್ಲ. ಇಫ್ ಯು ಹ್ಯಾವ್ ಅಂಡರ್ಸ್ಟುಡ್ ದ ಕಾನ್ಸೆಪ್ಟ್, ನಿಮ್ಗಾಗಿ ನಿಮ್ಗೆ ಅರ್ಥ ಆಗುತ್ತೆ ಏನ್ ಮಾಡಬೇಕು, ನೆಕ್ಸ್ಟ್ ಏನಿದೆ, ಏನ್ ಫಂಕ್ಷನ್ಸ್ ಇರುತ್ತೆ ಆ ಸ್ಟೇಜ್ದು, ಎವ್ರಿಥಿಂಗ್ ಯು ವಿಲ್ ಅಂಡರ್ಸ್ಟ್ಯಾಂಡ್. Please don't by heart anything. Try to understand. So metaphase, what you saw, that spindle fibers were formed, and those spindle fibers, with the help of centrioles, spindle fibers were formed. And what happened? Some rays were there like this also, which did not connect. Those are astral rays, radiating rays. But those got connected, we call them spindle fibers, right? Where they got attached? They got attached to the plate which is called kinetochore. So to the kinetochore only the spindle fiber attached in the metaphase stage. Now what happens in anaphase? See here, try to understand. Anaphase, here because it is mitosis, the centromere will split. So what happens in the anaphase is, I am showing it here only. Here if you see, the centromere will split during the anaphase. So now it is not metaphase. The centromere have split. I will show you how it looks. Okay. So because the centromere have split, you will able to see the chromosome with the separated chromatids like this. See, are you able to find out the difference? And also you will find what? You will find the spindle fiber like this. This is the start of anaphase. What happened here? What you can find out? The centromere splits in the start of the anaphase. So you can write that the main function of anaphase is centromere splits. Are you able to see here? Centromere have split, and you know that spindles are attached to what? Kinetochores. I am again and again focusing on this because this is a NEET question. They'll ask you, spindle fibers are attached to which region of the chromosome? Usually we people think that it is attached to centromere region. Yes, it is centromere. So you should be careful about that. It is kinetochore which actually gets attached to spindle fiber. Okay. Which region of the chromosome exactly? It is centromere, but what is present at the centromere is kinetochore. Which actually gets connected to spindle is kinetochore only. Okay. So this is the start of the anaphase, wherein the centromere will split. Clear? Now what will happen? You can see that spindle fibers are made up of what? They are made up of proteins. Which protein? Please will you comment it and tell me, because every time it's just I'm asking my question my own and I'm answering my own. So I want you people to comment and answer me. Spindle fibers are made up of which protein? Or you can tell me which is the cytoskeleton which helps in the formation of spindle fiber. You will comment it, okay? Then what will happen is that the spindle fiber becomes shortened. They start becoming shortened. When they are shortened, the length of the spindle fiber will go on decreasing. When the length becomes decreasing, the spindle fiber, because of this decrease in the length, the chromatids are pulled towards the pole. You can understand here. Kinetochore attached alva. Yadu spindle fiber. Idu adana pull madutte. When it pulls it back, what will happen? How it will look? We will see it here. Okay. This is what is happening in the anaphase. So you can see that chromatids, you will see they are started moving towards the, they started moving towards the polar region. So what is happening here? The arms of the chromatids, the arms you can see that they are towards the equator, and the centromere region is pulled towards the pole. This is early anaphase, how it looks. Now what will happen during the late anaphase? Okay, what will happen at the late anaphase? Means once the anaphase stage is about to complete, then how the chromosomes will actually look. So that we will see here. The late anaphase, you will be able to see wherein these are the spindle fibers. Okay. what you will see is that almost you will see the chromatids have moved towards the polar region. Here also the chromatids have moved towards the polar region. Are you understanding this? Ana means what? I told you Ana means towards the pole. So now where are you able to see the chromatids? Chromatids have moved towards the polar region. Here the anaphase stage is completed. So what actually happens in the anaphase? First thing is centromere splits. Centromere splits. And second thing what happens is, so chromatids move towards pole or polar region. I hope you people are not able to see properly, so I am just telling this once again. First thing what happens is centromere will split. Second thing what happens is chromatids move towards the polar region. Okay, I hope anaphase is clear. Prophase, what all happened? DNA got condensed into chromosome, nuclear envelope disintegrated, nucleolus also disintegrated, and spindle fiber started forming. Metaphase, what happened? Spindle got completely formed in the metaphase, chromosome got completely condensed during metaphase, and spindle got attached to the chromosomes, and chromosomes came at the center. They lined up at the center. What happened in the anaphase? The chromosomes which are attached, the centromere splitted like this, and slowly it is moving towards a pole in the anaphase region. Okay, so take down this much. We'll see what will happen in the telophase. Okay, we will see what will happen in the telophase region. So clear? So anaphase is also clear. So we will have this anaphase diagram to compare it to understand the telophase. So we will have this anaphase diagram here. This is anaphase. We saw that what happened during the anaphase, chromatids moved towards the pole like this, and we saw that spindle was there as usual. It was connected to both the chromatids. Okay, this is late anaphase. What I am showing now, this is late anaphase. Once the anaphase is complete, this is how the thing looks. Now we are talking about telophase. Telomere region we have already done in chromosome, which is telomere means what? End. Yes or no? In the same way, telophase is the end of karyokinesis. Once the telophase phase is done, karyokinesis completes. So what will happen in telophase? The chromatids, see one more thing. Telophase is reciprocal. This is very easy. Reciprocal of prophase. So one thing you have to do, either you have to understand prophase or you can understand telophase, because they are opposite. Whatever happened in the prophase, the same thing in the opposite manner is happening in the telophase. So now you can understand what happened. So along with that I'll explain also. This is reciprocal of prophase. What all will happen here? What happened in the prophase? DNA got condensed into chromosome. Then ulta, no, here. What will happen? The first stage, I'll rub this. Take down that it is reciprocal of prophase. And the stages, let us see now. The first thing what will happen is, now it is already chromosome. Now chromosome decondense. Why? To form thread-like DNA. There condensation happened, and here it is decondensation, wherein the chromosome will start forming what? It will start loosening up. So I'll show you what will happen here. Slowly you can see that instead of the chromosome, once they are formed at the center, you will see that they start forming, again they loosen up to form thread-like material. Same thing happens here also. Okay, like this. It will start forming thread-like material. And now this is not late anaphase. Now we are talking about telophase. Okay. So the first thing is chromosome decondensed. Chromosome decondensed. Now what had happened there? Spindle fiber were formed in the prophase. Now ulta. Spindle fiber formation. Now you will not be able to see any kind of spindle fiber here. Gone case. Okay. Now next thing what we studied there in prophase. Nuclear envelope disintegrated. Now what will happen? Nuclear envelope. So for second question, second thing is what? Spindle disintegrated. Now the third thing is nuclear envelope. Prophase ಅಲ್ಲಿ ಏನಾಗಿತ್ತು? Nuclear envelope disintegrate ಆಗಿತ್ತು. So now it is ulta. Nuclear envelope, what you can say? What is that word? Reappears. Reappears. So what you can see now, both the side nuclear envelope should come, no? Because both of them are what? They are chromosomes, they are DNA. So the nuclear envelope will start here also. Formation of nuclear envelope. Here also you will be able to see the formation of nuclear envelope. I'll just show you the cell little bit bigger. So what you can see here is that if this is the cell, after the telophase, after the anaphase, what all things are happening in telophase. I hope this much you understood. Once the nuclear envelope reappears, one more thing, what was gone in prophase, it will come back. What is that? The last one is nucleolus. Nucleolus reappears. So what you will see? You will see that nucleolus also reappear. This is what happens in the telophase region, telophase stage. What all happened here? First of all, chromosome decondensed to form chromatin or DNA, loosed DNA, and then spindle which were there disintegrated. Already chromosome have been towards a pole. So here now the new nuclear envelope will start because we have divided successfully the nucleus. Telophase is the end of karyokinesis. So nucleus is divided into two parts equally. Then nucleolus also will reappear. So in telophase region you will find within one cell how many nucleus you will find? There are two nucleus. Now you will not call it as nucleus, you will call it as nuclei. Yes or no? Two nuclei are found in the stage. Which stage? It is telophase stage. Clear? So this is about telophase and this is the structure of telophase. Please take down this much and we will complete with cytokinesis. It's not that difficult. Okay, so successfully we have completed what? We have completed mitosis. Yes, we have completed mitosis, but only one phase of mitosis, which is karyokinesis, and which was very, very important as well. So we have completed that. We will come to the last one. That is what? We will keep this diagram to compare cytokinesis. So once the telophase is done, what is this cytokinesis? Division of cytoplasm. You can see that till now what we did? Till now only the nucleus was divided. Now we have this cytoplasmic content which we have already replicated. Where? We have already replicated this. Where, Baba? Every replication but we already did na. G1 phase. So it was already replicated in G1 phase. All the nuclear cytoplasmic content was doubled in the G1 phase. Now it is the time to equally separate the contents. So what will happen at the cytokinesis phase? What will happen is that cytokinesis is division of cytoplasm. So equal amount of cytoplasm is divided. How? here, if it is a plant cell, you will see plate formation. If it is plant cell, how the cytoplasm divides? By cell plate formation. What is this cell plate? See, if it is a plant cell, you will see that slowly a plate formation will start like this, which will divide the cell into two. This is called cell plate. This is called cell plate. And cell plate formation is usually found in plant cell. Okay. Now, after this, what is this cell plate? We have already discussed this when we were discussing the start of this chapter, cell, the unit of life. There we have discussed that cell plate formation is nothing but, cell plates are nothing but the vesicles which have been produced by Golgi apparatus. Okay, the Golgi bodies, they produce some vesicles. Those vesicles will line up and they form a plate-like structure. Later on, what will happen? Now this acts as a cell plate. Later on, afterwards, it becomes very hard and this forms a structure called, now after later on it becomes middle lamella. Middle lamella. And then what will happen? Once it becomes middle lamella, there will be wall formation here also. The primary wall formation will start here. The same thing will start here also, primary wall formation. And then you will find that, so this is how the cell is divided into two cells. Clear? So you know that after mitosis, the cell will not move away. It is always joined. Same cells, only plate formation will form. And again this cell will start dividing into two, again plate formation. This is how it continues in plant cell. Okay. So what you can understand here is middle lamella, the cell plate which has formed, which will help in division of the cytokinesis in plant cell. And then it will form middle lamella later on, it becomes middle lamella. And what will develop with inside the middle lamella is the primary cell wall. Okay, this red colored one what I have done. So this is primary cell wall. Okay. Then after that, secondary cell wall, we have already done about that everything. We will not discuss it again. Okay. I hope it is clear. This diagram you have to take down for cytokinesis in plants. This is cytokinesis in plants. Take down and we will finish it up with animals. Okay, now coming to the last part wherein we are going to see how cytokinesis takes place in animals. So plant cell, it is cell plate formation and the diagram is here as well. We will see how it will take place in animal. Animal cell, we usually show it round. Yes or no? Now already we know that DNA has loosened up like this. This is telophase. I am showing you telophase, what has happened in the telophase of animal cell here. So nuclear envelope has started again growing. It is developing now. Nucleolus formation also is found like this. And then this spindle fiber have been disintegrated now. Now what we are understanding is, okay, cytoplasmic content is also equally distributed. We need to separate the cell now. How? What we will do now? In animal cell, there is one important peculiar thing which happens. That is called furrow formation. It is called furrow formation. Furrow formation. What actually happens is, because in animal cell we don't have any cell wall, no? Here, the plasma itself will start invaginating like this. The plasma will start invaginating like this. And this is called furrow. Slowly what will happen, okay? You can pause the video and take the diagram if you want, okay? Slowly what will happen is, this furrow will start deepening like this. Like this. Then what will happen? Still more deepen. Still more deepening, you can say like this. And finally what will happen, the furrow will touch each other. I mean the furrow will start deepening still more, and then it divides the cell into two cells. So this is cytokinesis in animal cells. How it will happen? With the help of furrow formation. Clear? So this completes the cytokinesis. Now this is one cell and this is one cell. Here also this is one cell and this is one cell. Now we have completed mitosis phase very easily. I hope you people have understood it easily. So please comment the answer which question which I had asked. Okay, and then next class we will see what is the use of this mitosis, and we will see overview of, or we will start with meiosis. Okay, take down.